Okay. Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 66th seminar, and I call it on Australia Day. It's already February. I cannot believe it, and time really flies. And I just started my first seminar trip this week. I look forward to meeting students, friends, colleagues, and researchers in person again all over the world. And I will reduce the number of my trips this year, not more than 160 days like last year, but it looks like already more than 100 days this year again. Okay, it's my tremendous honor to introduce very briefly our pioneer speaker, who is also traveling right now from Australia, Dr. Claudia Vickers. She is actually, very, famous, scientist in this, synthetic biology field, and she used to be a professor and an academic leader in Australia, leading synthetic biology efforts there. Recently, she moved to Eden Brew, an animal-free dairy company, as the chief scientific officer to focus on translational technology development. She is also an editor of ACS Synthetic Biology, one of the probably the famous, synthetic biology journal right now. What else do you want to know about her? So Claudia, I really thank you for your time and also for huge contribution to the scientific community. And then you also care about people, and that means because I will see it. You spend some time for us even during your conference trip in US right now. So the virtual podium is all yours, and thank you again.
Thanks very much, Tae-seok. I really appreciate that introduction. G'day to everybody. As Tae-seok said, I'm Australian. I'm actually New Zealand by birth, so kia ora to those of you who get to hear from New Zealand. I'm, I guess, a recovering academic, but I still have two academic positions. So I'm still an adjunct professor with the Queensland University of Technology and also with Griffith University. And I have a small research group at Queensland University of Technology, still working on metabolic engineering of natural products in Saccharomyces. But most of my time, in fact, a lot of very, very much of my time, more than I should be spending in extensive sleep, happens at my company, Eden Brew. So Eden Brew is focused on making milk proteins, and we're making six milk proteins, so 4 caseins and 2 whey proteins, and we are recreating milk from those. And the primary driver of that work is sustainability. And the reason that we're doing milk is because it happens on massive scale. And if you actually want to have an effect for sustainability, then you have to do something that happens on a massive scale. So it's a challenging project, a very challenging project, but not impossible. And the product that we make can massively decrease the carbon emissions, land use, and the water use. But that's enough about me and what I'm doing. As I was thinking about going into this, I was really thinking primarily about what it is that's most valuable that I can tell you for your careers and your lives sort of going forward. And I want to start just a couple of minutes of discussion around this by saying that there are many good scientists in the world, but there are very, very few great scientists in the world. And you know them when you meet them. And it's not because they've published hundreds of papers and they've got an H factor of whatever. It's because they know their scientific material inside out and they're passionate about it, and they can share that passion with other people. And in fact, in my experience, there are very, very few people who really do this effectively, and who lead other people effectively to do this, and who teach people that passion. And that's sort of a special ingredient that's, I think, required to have a fulfilling scientific career. And sort of on top of that, when you're seeking your scientific career and choosing where to go, I think that the environment that you're in is so much more important than the quality of the lab. And that environment depends mostly on the people. So when you're looking around for postdoctoral opportunities and tenure track opportunities, interview the people in the group as well as the professor, and see what they say about the professor, because that person's really important for your career and for your happiness. And if you're unhappy in the place that you're in, then it's not going to be very pleasant. And there are lots of different things you can do. And if you're interested, even if you're interested broadly or interested narrowly, there's still a lot of different places that you can go to do that thing. The other thing that I wanted to say is something like, 99.9 something % of people with a PhD will not become a professor. And our training programs train people to become professors. And I actually think it's morally bankrupt to train people to believe that they'll all become professors when there are really very few people who go through the qualification training process who will actually become a professor. But there are many, many other important, fulfilling roles. And one of the massive problems with our industry is that not only do we train people to expect to be professors, but we also train them that they're failures if they're not professors. And this is pretty tough, I think, in the industry, because, I've already said not many people really actually become professors. For those other career paths, they can be, important and fulfilling, but there are two absolutely key transferable skills that you need to have a fulfilling career path, whether you're going to be a professor or whether you're going to do any of the many other things that you can do with a PhD. And the first one is an ability to write. So an ability to write is absolutely critical in an academic career path, because it's your ability to write that determines your ability to communicate the science that you're doing and your ability to write grants and get money in. So the academic metrics are very black and white. Papers up, money in. As painful as it is. And that's almost, dissociated from why we do science, right? But that writing ability is absolutely critical. And that's a transferable skill that is absolutely critical in every other pathway that you will ever have in life. So if you don't like writing, or you're not good at it, then get good at it and learn to like it, learn to love it. So you should be writing all the time. You're not just writing up something when the piece of work is finished. You're writing and communicating, and including presenting like we are today, and doing posters and taking every opportunity you can to get out there, talk about your science, communicate your science, write your science. The second really important life skill for academia and for everything else is your people skills. So your ability to talk to people, your ability to stand up, to differentiate yourself from the sea of other people, and to advertise yourself and not just the science that you're doing. And again, I feel that if people who are looking to recruit people are only looking for the quality of science, or for the what people have done, their H factor in their papers, they're not going to build a very healthy cultural or scientific environment around them. They're probably going to build a brutally competitive environment with lots of socially maladjusted people, and that results in a lot of difficult interpersonal relationships. So you should be looking again for an environment that is looking for your people skills and your ability to interact effectively and healthily with the people around you as you grow your scientific career. So in terms of the opportunity to be successful, seek opportunities when they arise, take opportunities when they arise, make opportunities when they don't exist. And you do that by putting yourself forward, by talking to people, by meeting people. In this business, you need not just a mentor, but you need proponents, people who will champion your cause and put you forward and talk to other people about you. So what Tae-seok is doing here is amazing. It is very, very special and very, very important for, I think, the early career researchers like Sara, who are taking the opportunity to present in the series. So thank you very much for doing this. I will end my few minutes of introduction there. If you have questions or want to explore it further, my inbox is always open. Sometimes I don't respond as quickly as I like to, but I'll get around to it eventually. And just remember, you know, there's lots of people who are good, there's lots of people who are great, but the environment that you're in is the most important thing, and there needs to be nurturing.
Fantastic. You know what? That is actually the thing I actually giving to junior people when they ask for some advice, and you summarize beautifully. Thank you. I mean that, and you know, I 100% agree with you. I mean everything you said. I mean, actually, Sarah actually interviewing right now, in a multiple place, and the interview is actually they evaluate the candidate, but at the same time, as you mentioned, we want it to be in the place you will be happy, right? We will be happy, and then that means we also evaluate our potential colleague in the future. So that is good thing. But the keyword you multiple time emphasize, keyword in academia, I believe, should be not money, you know, not other thing, not infrastructure. The keyword should be people. Because at the end of the day, people make big impact, not the money, not the infrastructure. So that people somehow forget or ignore, but that is the most important thing, I believe. So that if people have that in mind, I believe this world will be much better place to live, especially academic field. So thank you so much. I mean, thank you. That's another thing, you know, I want to mention. You said professors should love writing. I remember now, you know, I need to give a keynote talk early in the morning, but I wake up early because I need to write a proposal and submit on that day. So I actually prepare whatever 3 hours to submit that proposal rather than preparing my keynote talk on that day. So constantly writing, that is one thing you should love. Otherwise, this job is always challenging. Okay, so thank you. Do you have anything other thing to add?
No, I didn't want to take up too much time. I'd rather listen to Sara.
Okay, thank you so much. Okay, so I will, in the case, you know, introduce the main speaker of today. Of course, with a longer, you know, remark introduction. Dr. Sara Molinari started her studies in Italy. Oh, I envy you. With a Bachelor of Science in Pharmaceutical Biotechnology at the University of Perugia and a Master of Science in Bioinformatics at the University of Milano Bicocca. She then joined the Systems, Synthetic, and Physical Biology PhD program at Rice University and graduated from Matt Bennett's lab with a thesis on programming differentiation in bacteria. This work enabled the creation of a novel kind of pattern formation by physically separating genetically distinct cells. Actually, that paper, one of the paper I love the most. As a postdoctoral researcher, she created the first macroscopic de novo engineered living material that grows from genetically engineered bacteria. As a future professor, as I said, she's, you know, interviewing multiple places. She will develop living materials able to interface with complex environment, such as the human body. She's in the job market, as I said, multiple times, And if any hiring people are watching this talk, please consider her as, I mean, the hiring, hire the person and give the offer. She's the real rising star, and I highly recommend her. And Sarah, good luck with your job search, and I believe you will get multiple offer. And thanks so much for your time today. And please take it away, and all yours.
Thank you, Tessa. Like as Claudia said, what you're doing here is really helpful. It's truly extraordinary. Your effort spans multiple years, and you help so many young people. So we're all extremely thankful for this opportunity. Thank you, Claudia, for the beautiful words. Like I'm actually really agree with what you said. And throughout my PhD and my postdoc, I could really understand how my luck was made mostly by my PI, were like not just like scientific mentor, but they're like life mentor that guide me through the process, and that were my first advocates. And I want to just take the opportunity to really thank Matt Bennett and Carolina Franklin for everything they've done for me during my career. And the reason why I'm here now interviewing for job is mostly and largely due to their effort as mentor and people. So I was extremely lucky with my mentors. And so now let me share my presentation. So today I'm here to talk about the novel engineered living materials that come from bacteria, which is like my major area of expertise. And I actually want to start with a brief introduction and say that actually I took like a weird path. I started from biology, like Tessa say, from pharmaceutical science, and then I transitioned from biology to engineering. And the reason why this transition happened is because I realized that really you can build with biology. And in fact, nature is the greatest architect ever, and it was able to create a multiple different functional structure. They're truly remarkable properties and characteristic. And so all my science has been inspired by how can we start from here and end up here. So what's the process to create this incredible functional architecture? And so what I'm trying to do right now here is to create materials from bacteria. And when we talk about materials, it's very easy to understand how they actually support human progress, because they mediate every single activity, daily activity of the humankind. And if you think about it, every time you work or do any recreational activity, you physically interact with man-made material that support the activity that you're doing. So designing the material of the future really means try to understand where the human progress is going and anticipate it. And so if we think about what are the characteristics that materials of the future should have, I would say the major thing that we're looking at is a superior performance. We want them to have some characteristic that current material don't have, so that we can enable a different application, especially at the interface with complex organisms like the human body. And here I'm referring to material for biomedicine, for example. But another characteristic that we really have to look for is sustainability, because we really can, like even Claudia said before, like we can start adding pollution to the world and adding waste to the pie that we already accumulated. And so this is very important, so that the new material are going to be sustainable. And another major point for me is that the new material have to be affordable, because we want to progress to actually be accessible for the largest number of people, and not just from a very restricted elite of rich countries. So how do we look for material that are performing, sustainable, and affordable? We really don't have to look far, because like I said before, nature already designed them for us. And in fact, living materials are surrounding us, and they're the most performing and sustainable material that we can think of. And when I'm referring to living materials, I'm talking about plants in all of their majestic diversity. I'm talking about the human body. All of our organs and tissues are living materials, but also biofilm, the desert crust, it's a living material, for example. And so despite their extraordinary diversity and multiple forms that they comes with, living materials always share a common architecture. They're made of living cells that control the biological function of the tissue, but they also produce a polymer matrix that embeds the cell, traps them in there, and it's ultimately responsible for the mechanical and physical properties of the material. And so the fact that this material have living cells gives them the hallmark of being multifunctional. So multifunctional because they have a unique combination of biological and mechanical property. And let me tell you that multifunctionality is a characteristic that is extremely difficult to engineer in materials, but it's also extremely transformative. And for that, I'll give you an example. So imagine if you have a wound dressing that not only mechanically protects the wound, but also prevents the formation of biofilm, stimulates tissue regeneration while also preventive excess scarring. That would be absolutely transformative. It's an area of very active research, and there's not an ultimate solution for it. In the same line, we could imagine having packaging, food packaging, that also reports on the presence of toxins, or clothes that inform on the status of human physiology. On top of that, this material also grows from cells. Like we said, the cells is responsible for creating also the matrix that embeds that. And so in this way, they really require minimal energy input, meaning basic nutrients and standard environmental condition. And in this way, they're also sustainable, but they're also very easy and cheap to fabricate. And so they become very affordable. So my idea for the future is to really being able to reproduce this biological process, engineer cells so they will self-assemble into whatever functional structure we will be able to design in the future. But where do we start from for this very ambitious challenge? So in my career, I actually help not only addressing some of the challenging, but also defining what are the needs for growing materials from the bottom up, meaning from the engineer cells up to the functional structure. And I define here some of the challenges that I define. I actually also brought it to the lab and found a solution for them. And today I'm going to talk about a few of this that I addressed throughout my careers. And the first is designing stable powder formation, which is something that I work on during my PhD. I also solved for reaching the macroscopic scale and found a way to encode tailored mechanical property into materials. This is something that I work on during my postdoc, and I'm going to briefly mention what is the potential for the future. So designing powder formation, why is that important in the realm of engineered living materials? It's because if we look at living materials here, for example, that's an example, that's a picture of a plant tissue, we'll see that cells are not all the same. So they're a collection of multiple cells, and it also not a random connection of multiple cells. I mean, our body is not like a random collection of cells, but they have a very specific order and they form very specific architecture, and that's actually what enable superior function. And so the first thing that we really want to achieve is the ability to differentiate the cells, so to achieve multiple cell types that then we can distribute in space in controlled fashion, so that we can enable stable powder formation that can be leveraged to increase the functionality of the population. So there have been previous efforts to distinguish the population, mostly based on gene expression, which is a very effective way, but it's also susceptible to noise. And if we are thinking about the activity of a three-dimensional structure, we're talking about every single cell needed to carry out a function for a longest period of time, days or even weeks. And so we would like that single cell to never be able to revert, to switch from a state or another. So we wanted that differentiation to be irreversible. And so what I imagine doing is having a division system that will ultimately create two genetically distinct daughter cells, a sort of asymmetric cell division. So I started from a basic plasmid system that this audience need probably no explanation. You know, when we leverage the ability of the plasmid to partition among symmetrically among the other cells, and we interfere with this process to engineer a system that would enable asymmetrical cell division. In this way, we try to trap all the plasmid in a single cluster, and we did it by leveraging the ability of this specific protein part B to bind to a sequence within the plasmid, but also to bind to itself. And so in this way, part B with this like multiple interaction ability will trap all the copies of the plasmid in a single cluster. And then it's easy to understand that when you have a unique body that contains all the plasmid, upon cell division, it can go only one way or the other, leaving one cell identical to the progenitor clone that created that, but the other one without the genetic information that was originally contained in the mother cells. And for that, this will be irreversibly differentiated. So we first show that actually this system worked, and here you can see the progress of a single cell that is forming this cluster of plasmid, and whenever it divides, this cell lost forever the ability to produce a red fluorescent protein, which is the information contained in this plasmid. And you see that the offspring that comes out of this cell slowly turns down the fluorescence, and it's never going to be able to reacquire it because it's lost that genetic information, while the mother cells always keep producing fluorescent protein because it kept the plasmid. And so, like I told you before, this is a very important because it enables stable pattern formation. And in fact, by tying motility to the presence of the plasmid, meaning that when the plasmid is present, the cells has its motility impaired, and when it loses this inhibition, becomes able to swim again through agar. And so here you'll see that when you spot cells on agar, you will keep a center of progenitor cells that never grows, and then a halo of differentiated cells that you can see here in yellow that are swimming away from the center of the colony. And so this actually was a pattern formation that was proved to be stable for multiple days, and we know that the cells inside this pattern will actually not reverse between a status and the other. So back to our challenges. Now I'm going to talk about my more recent work, the one that I've done in Caroline Auer's Franklin's lab, trying to engineer actually macroscopic structure from the bottom up. And so in order to achieve this goal, one very, very critical ability that we need to gain is to genetically reproduce the natural process where the cells self-produce their matrix that then embed them and confer the mechanical properties to the material. So in few words, is the ability to genetically encode a synthetic matrix that at the same time assemble cells on the macroscopic scale, but also give us control of the mechanical properties of the material. So there are multiple options for biopolymer matrices, and in fact, we've seen that a lot of microbial biofilm prefer carbohydrates-based matrices. There's also protein-based material that we can leverage, or biominerals, and all of them are part of the realm of matrices that we actually find in natural living materials. But from an engineering perspective, these polymers have very, very different characteristics, especially in the way we actually produce them. Protein-based materials are pretty standard. So the greatest advantage of protein is that the presence of protein production is not just standard across organisms, but it's more or less standard across different kinds of proteins. Carbohydrates and biominerals, on the other hand, have very specific pathways of reaction. They're very sugar specific, carbohydrate specific, or biomineral specific. And so re-engineering this pathway is extremely difficult compared to producing protein. And so this became the polymer of choice for the bottom-up engineering of living material. That doesn't mean that in the future we can also leverage different kind of polymer, but that was the easier starting point from an engineering perspective. So prior to my work, engineered living materials were either microscopic, and then they had to be processed into microscopic materials, or based on natural living materials like the nanocellulose, which is a really and truly phenomenal material, but it's also scarcely tunable. So it's hard to dramatically affect its mechanical properties. Or they were assembled top down, which gives like the most freedom in the kind of cells and the kind of matrices that we can mix up, but also the resulting material lacks the ability to self-assemble and self-repair, which is critical for affordability, sustainability, but also, most important, performance. So I engineer the first macroscopic engineer living materials, starting from Caulobacter, the freshwater bacterium Caulobacter crescensis, which is known for being covered by a surface, by a layer of protein. They're called S layer, surface layer. And here I want to stop for a second to thank Professor Kathleen Ryan from UC Berkeley, who is the absolute Caulobacter expert and taught me everything I know about working with Caulobacter. So the surface layer of Caulobacter is made of a single protein monomer called the RSA. And in fact, here you see different colors, but they're all identical copies of a single monomer that self-assemble into this hexagon structure, then cover the entire surface. This protein is made of a surface anchor that makes sure that the protein doesn't get lost in the medium, but actually stays attached to the surface. A crystallization domain that is responsible for the three-dimensional structure of the protein, and a very unusually big secretion domain that was also shown to self-aggregate. But in order for that to self-aggregate through hydrophobic interaction, it really needs to be free from this very rigid structure that is the native structure of the protein. And so with a single domain swap, I actually replaced the crystallization domain with an elastin-like polypeptide, which is a polypeptide derived from the human tropoelastin repetitive sequence. So what happened is that this elastin-like polypeptide is extremely unstructured, and so it unfolded the protein on the surface of the bacterium, leaving the C-terminus secretion tag able to self-interact. And so it's like creating a layer of sticky glue so that when cells come in contact with each other, stay attached to each other because of the sticky glue that they're both surrounded of. But at the same time, it also introduced an elastic structural material that, and as I'm going to show you after, is going to give me control over the mechanical properties of the final material. And so we first confirmed that we actually redesigned the surface of Caulobacter crescensis, and you can see here that this is the very regular lattice of the wild-type bacterium. And here is our strain that actually shows this brush-like structure where protein projects outside of the surface, which is exactly what we intended to do when we replace the crystallization domain with the ELP. We also show by staining the matrix with a spike catcher fluorescent protein and leveraging the presence of the spike tag, we show that the matrix surrounds every single cell like intended. And so now the only thing that's left is actually putting this strain to work and see if it forms macroscopic materials. And what I'm going to show you is the picture from the bottom of the culture flask. And as you can see, unlike the wild type that grows in a planktonic form, our de novo ELM strain really forms macroscopic aggregation of materials. We probe this material, we take picture of it, and we probe it with optical microscopy, and we've seen that it's made of large protein aggregates. And I want to point out that other people in the past have tried to aggregate cells with other displaying system, but they got amassed cells in the order of tens of hundreds, and here you see that we got much beyond that scale. And so this was very encouraging results. And another thing that I want to point out is that this experiment at the time of publication was repeated over 100 times. Probably now that number has tripled. The experiment has been performed by multiple scientists, including the material scientists at Berkeley that actually perform all the atomic force microscopy analysis. They were not trained biologists, and yet they had no problem growing the materials, and it never failed. We never fail seeing the formation of macroscopic materials after more or less 24 hours from the inoculation. So that means that this mutation actually does not affect dramatically the fitness of the cells, and the cells do not try to escape this mutation. So we also characterize the internal structure of the materials, and once again we've seen that the matrix surrounds every single cell. So it's kind of not surprising that it permeates the whole material. We can see that in blue the matrix and in yellow the cells. What we didn't expect to find, though, is that at intermediate level of magnification, you see these clumps of matrix, which are unusual because, again, the matrix was only intended to be on the surface of the cells. So what it means is that some of the protein actually gets released from the cell, gets secreted in the media, and form these clumps of matrix. We're trying to investigate further this phenomenon, and we've seen with atomic form microscopy, again with the help of research in Lawrence Berkeley National Lab, that at the beginning the planktonic cells have a pretty smooth surface, but as the material grows, even the single cell start forming bumps of on the cell surface. So we think that these bumps are actually made of our protein that layers up on the surface and eventually gets detached enough that gets ultimately secreted in the medium. And in fact, while investigating the presence of the protein in the media, we found that both in standing and shaking condition, there's some sort of release of protein. At this point, we know that the matrix plays a huge role in forming this structure that actually organizes the cell over 4 orders of magnitude, because we go from a micron of the single cell to a centimeter scales of the material. And so we would like to prove that changing the matrix would also reflect in a change of the mechanical properties of the material. And in fact, we produce 3 different strains, one that lacks entirely the elastin-like polypeptide, and one that lacks the surface anchor. So these are the main difference, is that these 2 materials are actually still displayed on the surface of the cells, but this protein is entirely secreted, like most of people have done before. It's a material that leaves the cells and is secreted in the media. I don't know how obvious it is from this picture that I selected, but this material actually looks different, like they have a different visuals. And under the scope, these two material actually looks pretty identical. It's really hard to distinguish them, and both are characterized by this large microscopic aggregation. But the third one, as you can see here, has a much lower amount of cells. Like you can actually see the matrix and just few cells. They're loosely attached to it. And so here we already got the first design rule. And in fact, if we want to have a cell-rich material, we really need to display, not only secrete the matrix, because this will allow for cell-cell interaction, but also for cell-matrix interaction, allowing the formation of a cell-rich material. We also took this material, put it under rheometer, and actually characterized as storage modules that gives a measure of the elasticity or the stiffness of the material, and the loss module that describe more the viscosity part of the material. And one thing that we found is that the storage module is constantly above the loss modulus. In fact, here there's two different Y scale. And this means that the material is a solid. And very interesting, the mechanical property from the stiffest to the softest material vary over 25-fold. This was absolutely unprecedented and extremely exciting, considering that it's only reached by genetically engineering the cells. We also try to give an interpretation of this result, and we think that the reason the material is the stiffest is because the ELP retains a lot of water, so it will lose water content when this material is absent. And on the other hand, the softest material is soft because by releasing the connection between the cells and the matrix, we actually decrease the amount of bonds that are within the structure of this material. This is though an area where we seek to grow and try to understand better of how like the internal structure of this material is ultimately responsible for the mechanical property of the ELM. We also characterize the formation of this material. So instead of just like inoculating, going to sleep and find the material the next day, we actually follow the growth of the material throughout the night and show that these cells first start growing plantonically, but then start accumulating a pellicle at the water-air interface, that then becomes thicker and thicker until it collapses into a unique piece of material after a few hours of the pellicle formation. And so what we understand is that by affecting the water-air interface surface, we were able to modulate the size of the pieces of material that form in the flask. And we narrowed down to these three major physical factors, that is the culture volume, the shaking speed, and the flask diameter. And actually, Bobby Tesorero, who's the second author of the paper, also the first PhD student that I mentor, narrowed down and created this mathematical model that is able to incorporate all these three parameters into a unique value, and identified ranges for which we can have the optimal size, like the largest size of material formation. And this model was also found to be predictive. And so here we provide a description of this phenomenon. And if you just think about shaking speed, for example, when it's too high, it's very difficult for the pellicle to form, and it will scatter into micro pellicles that then end up being like very little specks of material. If the shaking is too low, it fails to aggregate the material in the first place in a pellicle, and again is only resulting in few specks. But then in the optimal range, the process go as intended, and one single pellicle forms that forms a large piece of material. And this mathematical description is actually extremely useful, not just for the scale up for application, but also for the scale down for rapid prototyping of future materials. We also show that this material have very interesting characteristics. For example, it can be forgotten on the bench for up to 3 weeks and then reinoculated to form new materials, which is an extremely interesting characteristic, especially if you think about the fact that transporting materials is extremely difficult. Imagine the first vaccine that had to be stored -80. That was like a huge hindrance for distribution. So when we think about making therapeutics out of this material, this would be a great advantage. It's transportation and storage. They can also be processed into cohesive paste and used as cementing agent. One of the things that's really interesting is that this material actually retains the ability to filter cadmium. That is already a characteristic of the single cells, but we'll point out that because this material is a solid macroscopic material, it will make a much easier it will provide a much easier way to actually filter solution than a solution of dispersed cells. And so I like this application because it doesn't need to have further cell engineering to prove how ELMs can actually unleash the power of biology and make it macroscopic and useful. Because of the presence of the SynBYSS, we can also functionalize the material and use it as a catalysis. In this case, we provide a proof of principle with glucose dehydrogenase. So in summary, the thing that I really would like you to remember is that I created the first de novo macroscopic engineered living material that grows from engineered cells. It is held together by a synthetic protein-based matrix that not only assemble the cells on the macroscopic scale, but also by genetically engineering the matrix, we are able to control the final mechanical properties of the ELM. A very quick glimpse into the future. Now we have created a new class of biomaterials, and so how can we really tailor not only the mechanical property, but also the biological function to create multifunctional material. And that's the answer that I'm actually seeking to the question that I'm seeking to answer in the future is trying to engineer the cell and the matrix so that we can specifically tailor the property of this material for a specific application. And so with that, I actually want to skip over these few details and give you the perspective that this material actually can be tailored, can be particularly useful for application at the interface with the human body and with plants, because the ability of the cell of process multiple input and respond with biological function. They have the capability to cope with the complexity of multicellular organism. I'm gonna seek to incorporate expertise so we can actually characterize the structure of the internal materials and have a better understanding of how every component affects the final property of the material, and also incorporate rational design. So in the future, we can imagine a structure and know which core component we have to incorporate to reach the desired final output. So with that, I would like to really thank the group that supported my previous work. First, my PI, Caroline Franklin, and all the people in the lab and the people that directly contributed to the work that I presented. Also, our collaborators at Berkeley and UC Berkeley and the Lawrence Berkeley National Lab. Funding, of course, and with that, I'll be happy to take any question.
Fantastic. I love it. So I see many questions in the chat, but before going there, Claudia, do you have any question or comment? Okay, she may have some connection problem. She can come and go on somehow. So I'll start with some questions. So question here from Pravin. The first question is, what is the purpose of SynBYSS?
So the SynBYSS has a portion of functionalization. As I've seen you before, if you look at this picture here, we have stained the matrix in blue and the cells express fluorescent protein. So we see this blue because we actually incubate the material with a SPY catcher GFP. And so because here that the SPY tag, I'm able to stain specifically the matrix, and so understanding where in the outside of the cell I can find the protein. We also show that as you can attach not only fluorescent protein, but also active enzyme, for example. And here we'll see that the SPY tag provided a mean to functionalizing the material from the outside.
Okay, the second question from Pravin again is, what is the ELP60 and from where elastin-like polypeptide came from? The second question.
So this is an elastic-like polypeptide that we've worked with in the past. We obtained that from a different lab. It's a 60 repetition of the pentamer. It's a non-cross-linkable ELP. But if you want the sequence is actually published in the paper. But if you want any more information, I'd be happy to provide more detailed technical information about that.
Okay, I guess Claudia has some connection problems. So I will ask some a little bit big questions. So I love the, of course, I mean, I love the both project, but I'm a little biased. I love the first one more because that's more core genetic circuit thing. But my question is actually for the second part. So, people talking about, making even house out of living material, and probably that is not 5-year project, probably the decades, we probably need to work on multiple years, like 20 years or whatever, to achieve that goal. But another thing I fascinated by your talk is you're talking about at the end, but you couldn't go in depth, about utilizing this material for some human, related application. I just wanted to hear more about that part, because that is something I'm interested in working on in the future, but I haven't started.
So yeah, I think that this is the cadmium filtering solution. The cadmium filtering idea is actually what inspired. So Calyobacter crescentus is already able to filter cadmium, right? So you can incubate the cells with a cadmium contaminated solution, but then you have to centrifuge and take the single cell. Well, if you have a piece of material, you just put it in solution and take it out. So the idea of having a macroscopic structure would make the natural property of bacteria usable. It gives them in your hand, right? And so with that, you can imagine to use probiotics to make this material, which would then provide a much more reliable tool for colonizing the intestine, because they don't spread out, but actually they stay in high density. And you can imagine also adding layer of engineering on cellular and matrix part to make them even more strongly suitable for addressing specific diseases. You can also think about plant microbe interactions. They're very popular right now. There's a new call on that. And but the question number one is how you make this microbe persistent in the plant microenvironment, right? And so having them again organizing in a material in a paste can provide a solution to this question, because the cells is not only then persistent in the plant microenvironment, but it's also protected because this system provides a sort of encapsulation around the cell that is self-regenerating. That's also one of the things that attract me most about making materials that self-assembles, because they can keep on producing this capsule that will protect the cell from external damage. And so that's how we envision to actually leverage engineer living materials for new applications in the future.
Okay, that's wonderful. So thank you. In that case, my first kind of comment or question. So could you kind of share your perspective on living material potentially making house or living place?
Yes. So for now, for example, we've seen here that they can be semantic agents, right? So this is a first little step. Of course, the material after a long time is not living anymore in this case, but you imagine you can have a very cheap and ready usable glue to assemble other materials. So a hybrid combination of existing material and engineer living material could be the first step to reach really macroscopic scale. As in the meanwhile, we have to really understand what are the limitations of growing of assembling cell of a large scale. And we already, like I told you before during the talk, we already went from 100 cells to thousands of cells. So we already increased that scale by a lot. And so as we understand the design rule of this process, we can imagine going bigger and bigger. And with bacteria, we can really think big because, right, like a bacteria multiply at a very impressive scale. So they really have the potential to do macroscopic structure, but we need to understand what are the design rule of the assembly principles. And this is like one of the things that my lab will investigate.
Wonderful. So my quick question, and then I will ask another question from audience. I have already one question. So the material itself does not causing any immune response for potential human application?
It depends on the bacteria that you choose. For example, there's probiotics that will not cause any problem in the gut. And it depends on the matrix that we're using. I believe that for biomedical application, that could be a one-by-one case where we need to really screen using animal models and try to understand what is the interaction between this material and the immune responses. It's hard to predict a priori,
Sure.
which is another thing that my lab will investigate.
Sure, wonderful. So another question is could this material be potentially used as a scaffold for lab-grown meat?
Definitely. One of the things that I'm extremely interested, for example, is like, we talked about organoids and scaffold for organoids, how the mechanical Q actually shape the differentiation of stem cells or the organoids. And so this material can provide not only mechanical cues, but also biological cues. So can we use them to shape the growth of mammalian cells? That's and so lab grown meat, for example, that's a very good question.
Okay, so actually I have another question that is actually asked earlier about ChatGPT. We answer actually in the chat, but Claudia actually should go. I mean, so she left, so she gave some answer. My sort of answer to that one is, as an educator, I'm a little bit concerning, and then I also posted multiple my opinion in the social media. But at the end of the day, as a researcher, I mean, let me go from this way. I mean, so we learn language, new language. I mean, I'm not native speaker in terms of English, but I learn English, and I also saw my daughter becoming bilingual. But the important thing is language is not just kind of writing something to express idea. Language is also the way to understand the different culture. So for example, if I write the Korean, I completely structure the paper in a different way. That is the more logical to Korean, but not probably to the American or European people. So using that AI tools much easier, but at the end of the day, you also need to train yourself, to communicate with the English-speaking people. So in that sense, I not that much recommend using that AI tool for really writing something. but that might be good for getting some information quickly. That is probably one thing. Also for fun. I mean, people enjoy a lot with that thing. I mean, but at the end of the day, you know, just using that one, and also nature and science already express clear idea about using that, you know, algorithm, and they actually not that much, they against it, against using it. So I mean, that is kind of my answer. And do you have any, you know, comment on that one, Sarah? Probably that's a potential your question from your employer. I mean, because you know, they are all professors, they are worried about a lot. So
I have to say that I'm really not being an expert of this subject made me really careful about using it. It's because when you don't know something, when you don't master something, it's risky to adopt it without knowing what's behind. And so I feel that original work, like you said, for now is not really replaceable. I do see that becoming a tool that we can use in the future. Maybe after we wrote a paper, you can say, hey, can you write me the abstract based on what I wrote in the paper? So I know that it's going to become part of our daily life, but we need to educate ourselves and really understand to use it in a way that supports science and not trying to scam it or you know, cut the way around actually a solid honest body of work.
Yeah, I mean, you know, using it is would be, I mean, okay if that is not really publishing. Of course, I mean, even writing abstract, I mean, I also still believe, you know, summarizing our own idea in very short form, that's very important, because that's something like you giving elevator pitch. And then imagine you somehow met the Bill Gates, it's just, you know, by accident in elevator, and then you have that 1 minute to summarize your idea. And then if you successfully done, I mean, you may get $10 million from him. If you screw up, I mean, you cannot. So that's about the summary or abstract, and that's probably the most important piece why you rely on machine. I mean, we rely on multiple things, including Google search, many things, computers, so that changed our life. But still, you know, thinking is the most important part to me, I guess.
I agree with what you said, because it's like Google Translator right now. Google Translator is pretty phenomenal.
Yeah.
And you can really translate in any language and have an idea of what it says. But like you said, if I meet an English person and I don't speak English, I cannot like type on my phone, hey, you know, like it's something that you can use in to facilitate with your work, but it's not something that can entirely replace your skill.
No, I would, okay, I mean, you train yourself by looking at the abstract that is generated by the machine, and then try to improve your thinking process. But if you're thinking about completely replace your writing with that one, that is kind of a little bit dangerous kind of idea, I guess.
Agree.
Okay, let me double check whether we have another question. Okay, so we don't have more, and then I'm now see almost 7 minutes past the 11. So let me close. So thank all for joining and staying today, and we'll meet again on February 9, Thursday, the same time, the same journey. We'll have Dr. Hannah El Samad from Altos Lab and Professor Roland Hutchinson-Pickler from Montana State University. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers. I will promote you to panelists who can speak and show your handsome and pretty face if you wish. And thanks. I'm stop recording. Just give me 1 second.